little bit about what proteins are and why they're super cool, I'm going to actually put this in the context of a disease that many of you have probably heard of, Alzheimer's disease. So this is going to be a tale of two proteins that Emma just introduced, and I'm going to show you guys how they come to play and what happens when protein folding goes wrong. So our story starts over 100 years ago with this man here. This is Dr. Alois Alzheimer, and he noticed actually that many of his patients had memory loss, especially as they aged. When they got older, this memory loss would get worse. And after some of his patients passed away, their families actually donated the brains to science, and he was able to look at these brains and stain them for proteins, and this is what he saw. He looked at these brains and he saw particular clumps that he called plaques and tangles. Now to you, this might just look like a mishmash of different colors, but for Dr. Alzheimer, he was used to looking at a lot of these different stains, and so he realized that these were very different from what a normal brain looked like. And I'm just showing you guys here, this is what the normal brain looks like, and there aren't these big clumps of little dots, only these little round circles. And so this made him think that, okay, something different is going on in these Alzheimer brains, or in these brains that he defined as having Alzheimer's. And he wanted to study this and really understand what is going wrong. So as you guys probably have deduced, Alzheimer's disease is based on this finding, and it was defined in 1906. So that was a really long time ago. So what do we know today? How much have we learned? Well, I'm just going to start with a brief overview of what the symptoms are and a basic clinical picture of what Alzheimer's look like. So we call Alzheimer's a neurodegenerative disease, and basically what that means is that neurons are wearing away or degenerating. And so in this image here, you have a healthy brain compared to the brain with Alzheimer's disease. And you can see the difference very clearly. The brain with Alzheimer's has a lot of holes in it, and you can see that the parts seem to be wearing away or dying off. And remember, a neuron is a cell that makes up your brain, and this is just an image that Emma showed earlier. And so a lot of you know actually that Alzheimer's is associated with memory loss, but there are a couple other symptoms that I just wanted to present to you. So one of them actually is confusion with the time and the place. So some people with Alzheimer's have difficulty remembering what date it is, what year it is, and they also have trouble finding their way home or trying to think about where they are in the world. And this can get worse as the disease progresses. Another symptom is just basic forgetfulness. So they will forget where they put their keys, or they will forget to water a plant. One of the first symptoms that arises is a difficulty finding words. So some people will have trouble starting a conversation or putting words to different objects around them. And this can also get worse as the disease progresses. And then finally, as the disease progresses, it gets harder and harder to complete familiar tasks, whether that's paying a phone bill to ultimately even brushing your teeth or making food for yourself. And so you can see how Alzheimer's can be a really devastating disorder. So what can we do about it? Well, let's go back to this image and figure out what's going on with these plaques and tangles and how proteins can actually help us learn about Alzheimer's and maybe even solve the problem. All right, so our first question is, what are these plaques and tangles? Well, we actually know that these are misfolded proteins. So what is a misfolded protein? Remember, Emma told you guys that a protein's shape determines its signature dance move. So if a protein doesn't have the right shape, it can't do its function, and it can't do its dance move. And this is what we call misfolding. So just like in origami, where you have to fold something in a particular way in order to get the product, the same thing happens in proteins, where if you don't fold the protein in all the proper steps, you might just get something that looks like a clump or like this piece of paper. And so again, if you have a misfolded protein, it can't do its signature dance move, and that can cause difficult things in the cell. So how do we actually misfold a protein? Well, remember this timeline that Emma talked about, how to make a protein, starting from your DNA, going through your RNA, amino acids, modifying those amino acids, and finally getting the finished product. Well, actually, disrupting any of these steps can lead to a misfolded protein. Now, I'm just going to talk about 3 different steps, and specifically in the context of Alzheimer's, but it's good to think about how complex this process really is. So the first thing that can happen is a change in your DNA, or a change in your genes. And genes are just a unit of DNA that codes for a particular protein. So normally, your DNA specifies the order of amino acids, and ultimately that order will cause a normal protein. But if there's a change in your DNA, that can either cause an abnormal protein to be made, or if the change is really, really bad and the cell can't read that anymore, it will cause no protein at all. So this can be very dangerous. Another thing that can go wrong is when you're modifying those amino acids by adding the extra molecules to them, the body sometimes adds the wrong molecules. And when this happens, you can imagine how an amino acid change can become bulky in a particular area, and that will no longer be able to fold in the right way. So this is another important way that misfolding happens. And then finally, the other way that we talked about to modify proteins is to cut them, but sometimes the body doesn't cut the protein in the right way. And if you cut the amino acid chain or the protein in a different location, it can fold differently and ultimately can't do its signature dance move. All right, so keep these things in mind, and we'll see how this comes up again in Alzheimer's disease. So now that we know how proteins misfold, what happens when they misfold? How is this actually affecting the body? So we can think about this looking at this GIF of a marching band. So when one protein misfolds, it can't do its signature dance move, and so it's going to bump into other proteins around it that are just trying to do their thing. And ultimately, that's going to cause a disruption. And here, all of these band members are falling on top of each other and can't play their music. So similarly, the misfolded proteins are going to bump into each other. And what's unique about proteins is that when they bump into each other, they're not just like people who can get up and continue. They actually stick together. They're really sticky. And so this will cause just a sticky growing clump. And that's what we saw actually on that image before. So this is just an image of what a protein looks like when we visualize it in science, and you can see how the proteins can line up and stick to each other and basically make a stack like this. So the body actually has a cool mechanism where it can go through and sweep away some clumps that have formed. And it's important to note actually that clumps and misfolding protein actually happens randomly in the body. So sometimes the body just has too many amino acids, too many things to do, and that causes just a few of the proteins to misfold. And so it's good for the body to have this process because then it can clear away the clumps before they become too big and before they started disrupting other processes in the cell. However, as you get older, this process actually doesn't work as well. And so many people actually get more clumps of proteins as they grow older. And you can imagine how this will become relevant when we talk about Alzheimer's disease. Okay, so now stepping back, going back to Alzheimer's and trying to understand what's happening at a protein level. So what happens is that two proteins are misfolded and then they stick to each other. They can't do their dance. Now let's break that down. So this is just a schematic of what we saw earlier, of those plaques and tangles in relation to a neuron. And you can see that amyloid plaques are formed outside the cell and tau tangles are formed inside the cell. So what's amyloid and tau? Well, these are actually the names of the types of protein that are folding these clumps. So beta amyloid forms plaques and tau forms tangles. Let's talk first about beta amyloid and see what it looks like, what it does, and why it misfolds based on what we know about proteins. So beta amyloid actually comes from a larger protein. This larger protein is appropriately called amyloid precursor protein. And amyloid precursor protein is important in neurons because it helps neurons grow, and it also helps them communicate with each other. And you can see it here. It's actually located on the membrane of the cell, which divides the inside of the cell from the outside of the cell. beta amyloid is this part in yellow, and currently beta amyloid isn't itself a protein because it is part of this larger protein. Now, what happens in the body, and this happens randomly, so we don't actually know exactly why it happens, and it happens in everybody to some extent. The body goes and it cuts off amyloid precursor protein, and it gets rid of this top part and releases beta amyloid. And that beta amyloid is then released outside the cell, and because it's very sticky and it's misfolded, it's going to stick to itself. And over time, it's going to grow and create a clump that we know and that we've seen as a plaque. So this is what it looks like in a more schematic version. As these clumps grow in Alzheimer's disease, they take up a lot of space, and they're all formed by just that one beta amyloid protein. So this is bad for two reasons. First of all, it cuts off the amyloid precursor protein, which, remember, was important for neurons to grow and to communicate. It's also bad because you can see this clump really takes a lot of space, and it also blocks the connections between neurons and can also help them die, or that's our hypothesis. Okay, so now we know a little bit about beta amyloid, how it forms plaques. How about tau? How does tau form tangles? So tau is normally used to stabilize microtubules. So remember, Emma told you guys a little bit about microtubules. So that's this protein here. The microtubule is important to keep the neuron long and rigid. It provides structure, and it also acts as a highway. So other molecules can be shuttled along it to either end of the cell. And in a healthy cell, in a healthy microtubule, tau is actually going to stick to the sides of the microtubule and keep it in place so that it doesn't fall apart. But sometimes there's a modification to tau, and that causes it to fall off the microtubule and to misfold. And ultimately, this will stick to itself and cause a tangle of tau proteins. So what is this modification? Well, remember, I talked about how different molecules can be added to amino acids. And in the case of tau, extra molecules, shown here with these red circles, are added to the tau. And because these are extra bulky, the tau can't fold properly, can't interact with microtubules, it falls off, and it's going to interact with itself because it's super sticky and form these tau tangles. And again, if it's not cleared right away, if it's allowed to accumulate, then more are going to join in, and ultimately that's going to be really bad for the cell. First of all, because it takes up a lot of space, but also because microtubules then are going to be falling apart. So you won't have that structure, and you won't have a highway in your cells again. So you can imagine that that's not good. Okay, so now we know about these two proteins that are misfolding and can't do their dance. So how exactly do we integrate this, and how do we bring it together with why neurons degenerate? So this is just a fun comic. There are a lot of words on this slide, but basically it's just showing how having these plaques is going to disrupt neuron growth, neuron communication, and neuron stability, and that's going to cause them to degenerate or to die off, which we talked about earlier. All right, so this is really important to know about Alzheimer's, that it's related to two proteins. However, there are a lot of questions that researchers are still thinking about, and that a long way to go, actually, in order to make treatments out of what we know. So here's just some food for thought, some questions that researchers are thinking about right now. So we have some idea of how these proteins might be causing neuron death, but to what extent are they actually causing the symptoms that we see? So remember, correlation is not necessarily causation. So just because people with Alzheimer's might have these proteins does not necessarily mean that they're causing Alzheimer's. So there could be something else going on that is ultimately influencing the symptoms as well. We have a hypothesis, but we're still testing it now to really dig deep and understand the root cause of Alzheimer's. Another question is, I told you guys that these accumulations of proteins happen randomly. And so what predisposes some people to get Alzheimer's disease and not others? Why do some people just not clear away the protein as well? Is it more likely that the protein will misfold in some people and not others? And this is another really important area of research. So I told you guys also about two proteins. And is one of them more important than the other? What happens if you just have tau tangles, or if you just have beta amyloid plaques? And this is actually another controversial field in neuroscience, and there are even different camps that think that tau is the best, or the most important in Alzheimer's, and the one that we have to tackle, or that beta amyloid is the one that is really driving Alzheimer's. So this is another important question that researchers are now answering. And finally, can we stop tangle and plaque formation? And this is ultimately where a lot of treatments are trying to come from. And let's actually talk a little bit more about this, because there are some exciting developments that, based on what we know about protein misfolding, we can actually make better diagnoses and treatments. So one thing that we can do is, and that researchers have actually already done and are working on right now, is to make drugs that stop tangle and plaque formation. So we know that beta amyloid is caused by cutting, so we can actually make drugs that target specifically where proteins are cut and prevent that from happening. And so that's really exciting, and hopefully we might have some drugs on the market based on this, this research and these ideas. Similarly, we can also do the same thing to stop tau modification, so to block those molecules from attaching to tau and to prevent that. And that's also another exciting place where new drug therapies are being generated. I also talked about this, the way that the body sweeps away these proteins. And I don't really have much time to talk about the specific mechanism of this. It's related to the immune system, if you guys want to look it up later or think more about it. But basically, this is another exciting place where new drugs are being developed to bolster this process so that you can actually sweep away the clumps that are normally made. And then finally, using what we know about beta amyloid, we can find a way to diagnose Alzheimer's earlier. So today, we actually diagnose Alzheimer's really based on clinical symptoms. So if somebody is feeling like they're forgetting a lot, if they aren't really functioning that well, then they're going to come in and we can diagnose them. However, many times this actually happens pretty late in the disease process. And so if we can actually diagnose people earlier, maybe we can start treatments earlier, and that might actually help people regain their function or not to have as much memory loss as if we started treatments later. And so this is just an example of a way to image the brain, specifically looking for beta amyloid. So beta amyloid is shown in red here, and so if you put a person in a scanner, you can give them a molecule that's going to tag specifically beta amyloid. And if it shows up red, it means that they have a lot of beta amyloid in the brain. And based on this, we can say, oh, this person probably has Alzheimer's disease, and this will show up much earlier than a lot of symptoms. So we can start treating them earlier, which is important. Great. So in conclusion, what did we learn today? Well, first we learned about protein misfolding and how if you misfold a protein, it can't do its signature dance move. We learned where that misfolding can take place based on the process of making a protein. We also learned about what happens when you have a misfolded protein and why that can be bad for the body. And finally, we put this in the context of Alzheimer's disease and these two proteins, tau and beta amyloid. And so I hope you guys have been able to think a little bit with me about how we can use what we know about proteins in a way to help patients, specifically with Alzheimer's disease. Okay, so now we're going to hear from Lauren about hormones and what happens when proteins travel large distances.